Another thing is also one more thing, then after I will stop this one, right? Complete or incomplete linkage. Sometime it also asks complete linkage and incomplete linkage. Complete means it is very rare. Complete means what? Here only parental combination is there. Incomplete means it is normal. Incomplete means here not variations occur, right? Here two types of linkage. Two types is what? First one is complete linkage and second one is called incomplete linkage. Okay. Complete, I tell you, complete means what? It is parental. It shows parental combination. I tell you here because it is not given in NCERT, but they helps to clear the concepts, right? Shows parental combination as for example capital A, capital B or small a, small b. These are the parental combination, dominant and recessive simply. So they are rare in nature. They are rare in nature. As for example male Drosophila. Male Drosophila. In case of male Drosophila, because male Drosophila crossing over is not happens, right? That's why this is the example of complete linkage. Only parental combination is present, right? Incomplete, this one is very common, right? Male Drosophila, and another example is female. We can say female, male, female both. It is present in both. Silk moth. Right? Incomplete, it is common. Means what happens here? Here, due to the crossing over, here variation occurs. Variation occurs. Means very different type of combination we can see here. Different combination. Different combination happens here, right? So this one is called incomplete, right? Now here, now what is the significance of linkage? Why we are reading linkage, right? So what are the significance or what are, okay, before significance, what are the application of linkage here? Application of linkage. Because we know here linkage, strength of linkage means what happens? It is inversely proportional to distance, right? We can say here linkage is inversely proportional to distance, or we can say here also crossing over. Simply we said distance, here distance. Distance means what here? Distance is represented by percentage, that is percent, right? And this percent, it is called as centimorgans. This one is called as CENTS. Centi means what? Centi means 100, right? Percent means what? Percent means 1 in 100. That is centimorgan. It is the unit of measurement of distance. Unit of distance of link. linkage gene, centimorgan, and it is represented by percent. Do you understand? Suppose that I am saying 10%, 10% means 10 distance is there. Do you understand here? Okay, now come to the, now we come back to our NCERT. Linkage and recombination. Recombination means once in crossing over, what happens? They get separate. After separation, once more they recombine. That is called recombination. Simply here, Morgan carried out several dihybrid crosses in dihybrid. Already we know what is dihybrid, and I understand you know this one, right? Dihybrid, what is given? Dihybrid cross in Drosophila to study the gene that were sex linked, right? As for example, color of eyes, right? White and red eyes, right? These crosses, the crosses were similar to dihybrid crosses carried out by Mendel's in peas, right? For example, Morgan hybridized a yellow bodies, another way, because many variations are also there. Color of eye, length of wings, right? Color of body is there. It is given hybridized yellow bodied white eyed female. You understand here? Female means their body is yellow and their eyes are white. Two brown bodies and red eyes female, and intercrossed their F1 progeny, right? He observed that the two genes did not segregate independently. These two genes, they did not segregate independently, right? Now for each other, and in F2 ratio deviated very significantly from 9:3:3:1. This 9:3:3:1, it is obtained in case of peas, but it is not obtained in case of Drosophila. It is different, right? That is given. F2 ratio deviated very significantly from 9:3:3:1. Mendel get these ratio in F2 generation when Mendel means crossed a dihybrid cross, right, in peas, right? So he get that phenotype ratio 9:3:3:1, but when Morgan crossed this in Drosophila, he did not get this ratio, right? Expected when the two gametes are independent. Why? Because here we understand that male crossing over is very less. Okay, Morgan and his group, right, knew that the genes were located on X chromosome, right, and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion ratio of parental gene combination were much higher than the non-parental type. The ratio of parental gene combination were much higher than the non-parental type, right? Why? Because they are very close to each other due to the strength of link. So Morgan attributed this due to the physical association or linkage of two genes and coined the term linkage. Then Morgan he coined the term linkage. What he said, it is due to the physical association. Physical association means attachment of gene in a chromosome, right, or linkage of two genes, and coined the term linkage. Link means joining, meeting, right? To describe this physical association of genes on a chromosome, and the term recombination. So you understand term linkage means what? It describes the physical association of gene with chromosome is called linkage according to Morgan, right? And what is recombination? To describe the generation of non-parental gene combination, he used these two terms, linkage and recombination, right? Do you understand here? To describe the physical association of gene, he used term linkage, right? And the term recombination to describe the generation of non-parental gene combination. Recombination means what? Non-parental combination. What is given? Recombination to describe the generation of non-parental gene combination. He used two terms. Which one? Recombination and linkage. I think you understand what is linkage. Linkage means physical association of gene to a chromosome, right? And recombination means that is generation, right? Association of gene, that is describe the generation of non-parental recombination. Recombination means once they recombine. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked, very tightly or very closely linked. Tightly means what? They closely linked, that means tightly linked, right? Showed very low recombination because tightly linked. Tightly linked means distance between these two genes is very less, right? So crossing over is not possible. No crossing over means no exchange, right? Figure 4.11 is given here, cross A, and figure 4.11 in cross B. It is given, and through this figure we can understand, right, this means tightly linked or loosely linked genes and how they segregate, in which more segregation possibility is there. Very low combination, figure number 4.11 cross A, while other were loosely linked. Loosely means distance is more, then showed higher recombination. Recombination means crossing. After crossing over, what happens? They recombine with each other. Cross B. For example, he found that genes white and yellow were very tightly linked because the distance between these white and yellow is less, right? And shows only 1.3%. How many? Only 1.3% recombination is there, means very less recombination is there. While white and miniature wing, because white and miniature wing, their genes, they are located in a, means we can say a far distance is there, more distance, right, to a more distance part. The more distance means what? Linkage strength is weak, so here recombination possibility is more. It shows 37.2, much more than 1.3% recombination. His student, what is the student of this one? His people, Alfred Sturtevant, right, used the frequency of recombination between gene pair on the same chromosome as a measure of distance between genes, and also mapped their position on chromosome. He used as a measure of the distance between the genes, right, and he mapped their position on a chromosome. And today genetic map, the measure of the distance means what? How could we measure this distance? Through centimorgan. Centimorgan means 1 in 100. Do you understand? That's why here percentage also means we can say percentage also a measurement of distance. Here only for genes, not for mathematics. Are extensively used as the starting point in the sequencing of whole genomes, as we done in the case of human genome sequencing project. Human genome sequencing project already means not already, but means that the coming chapter we will read this one, right? So human genome sequencing according to distance. Distance means more distance is there, means more possibility of crossing over is there, right? Here this sequence also we use the genetic map, right, for human genome. This is described later. Now here this figure we can understand cross A and cross B. Here something you have to see here, right? You can see W and that is yellow and W. W means what? Here W means we can say white-eyed, right? So W means here their distance. See this distance. This distance is less. The distance between these two is less. Distance between these two is less. But when we see this one W and miniature wings, miniature wings, white-eyed and miniature wings, this distance is more, right? When we compare the distance between these two genes and in between these two genes, here we understand this distance is more distance. More distance means more possibility of crossing over is high. Possibility of crossing over is high. Less distance means possibility of crossing over is low. Less distance, it is less distance, right? LESSDISTANC. Less distance means what? Crossing over possibility is low. That's why here in F1 generation we get this one type, but when we see the recombination types, it is only 1.3, very less. Parental means 98.7 because their distance is very low, means here linkage possibility, here we can say crossing over possibility is very less, right? But here because distance is more, so that means we can, the strength of linkage is less, that's why crossing over possibility is high, and that means what here recombination type is also high. 37.2% means high, here 1.22% means very low. So do you understand? Means we can say here if distance is more means linkage strength is less, and then possibility of crossing over or recombination is also high. More distance, more possibility of recombination. Less distance, less possibility of recombination. Do you understand why? Because less possibility of crossing over, right? Why less possibility? Because less distance means what? It more strength of linkage, and here crossing over possibility is less. Okay, that is given by this figure. F1 generation, then F2 generation. F2 generation we also this one. Here you linkage result of two dihybrid crosses conducted by Morgan. Cross A shows crossing between gene Y and W, yellow and yellow body and white eyes. Cross B shows crossing between W and M, miniature wings and white. Here dominant wild type alleles are represented by positive sign and superscript, right? Note the strength of linkage between Y and W is higher than the W and M. Y strength of linkage is higher here because distance is less. Why here low? Because strength is Y strength is low because distance is high. The simply only this figure is given, so you need not to go very deep. Only one thing is there, distance more means strength less, and that's why recombination high, right? Here what is given? Distance less, that means strength more, that is strength of linkage between Y and W is higher. Why higher? Why strength is more? Because distance is less. Strength more means what? Linkage possibility is less. Crossing over possibility is less. Crossing over possibility less means recombination is also less here, 1.3 only. And here what is there? It is 37.2. Okay. Now a given here polygenic inheritance. Mendel studies mainly describe those traits that have distinct alternate form, such a flower color which are either purple or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, I think it is I already tell you this one. In humans, we don't just have tall or short people as two distinct alternatives, but a whole range of possible height is there, right? Such traits are generally controlled by 3 or more genes. So traits, thus they are called as these traits which are controlled by 3 or more genes, they are called as polygenic traits, right? Such traits are generally controlled by 3 or more genes and thus are called as polygenic traits. Poly means what? More than 2, and genic means genes, and trait means what? Characteristics or properties. Beside the involvement of multiple genes, polygenic inheritance also take into account the influence of environment. Human skin color is another classical example for this one. Height, and this one is human skin. In a polygenic trait of phenotype reflect the contribution of each allele. That means what? The effect of each allele is additive. Additive means what? We know dominant character number, it is totally dependent. That is, suppose that skin is there. So darkness skin, darkness means we can say it is controlled by 3 genes A, B, C, right. So 3 genes means A, B, C. Dominant character means dark character. So darkness of skin is depend upon the number of dark means what? Dominant character present in this trait, right. 5 dominant capital A, capital B, capital B, capital B, capital C and small c is there. That means this is more dark, more brown, right. Number of recessive genes are more, that means skin is fairer. That is called additive, right. To understand this better, let us assume that the 3 genes A, B, C which control the skin color in human with the dominant form A, B, C responsible for the dark skin. Dark skin is dominant over that is white skin or fair skin, and the recessive form A, B, C for the light skin color. The genotype which all the dominant alleles A, B, B, C, C will have the darkest skin color, negroid, and that which all the recessive alleles this will have the lightest skin color. As expected, the genotype with 3 dominant alleles and 3 recessive alleles, they have an intermediate color, that is called mulatto, right. Intermediate skin color. In this manner, the number of each type of allele in the genotype would determine the darkness or lightness of the skin in an individual. So do you understand this one? Means here number of dominant is more, means more darkness. Number of lightest means recessive trait is more, that means more lightness, more light skin is there, right. So this one is called as polygenic inheritance. Here I can tell you with this one. I think I tell you this one, but polygenic inheritance, what happens here? How could we? Here we have 2. First gene and second gene, right. In F1 generation means what happens here? We understand here capital A, small a, capital B, small b, capital C, small c. That is mulatto. Color, intermediate color, we can say intermediate skin color, right. A, B, B, C, C. And now what happens here? Here we have different means what? Here we get how many types? 7 types of colors, 7, and different ratio is also there. We can see here different means 1, 6, 15, 20, 15, 6, 1, right. Different means what? Here we can say capital A, capital A, capital B, capital B, capital C, capital C. This time what happens? Capital A, capital A, capital B, capital B, capital C, small c. So one is small, and this is capital A, capital A, capital B, small b, capital C, small c. 15. Third means what? Intermediate, capital A, small a, capital B, small b, capital C, small c is there. That is mulatto. Here what happens? Capital A, small a, capital B, small b, small c, small c. Here capital A, small b, small b, small c, small c, and here small a, small b, small b, small c, small c. We can say we have 6 combination is there, 6 types of combination. 7 type of combination, right. 7 type of combinations are there, right. So this one is darkest skin, this one we can say very dark but not darkest, light dark. This one is intermediate, or we can say mulatto, right? This one is light, this one is also light dark, right? And this one is very light, and this one is light test skin, right? So this one is called, we can say it is F1 generation simply, right? So this means what? It is totally depend upon the number of genes means what? You can see here number of dominant characters, now dominant genes we can say. How many numbers are there? 6 here, 5 here, 4 here, 3, right? 2, 1, 0. So when we see the number of dominant traits, so you understand all the traits are dominant means what happened? Darkest. 5 traits very dark, 4 traits light dark, half 3 trait means 3 dominant, 3 recessive means intermediate, 2 dominant, all other recessive means light, only 1 dominant, all other recessive means very light, and all are recessive means what? No melanin is there, lightest skin. So do you understand? It is totally depend upon the number of genes, number of factors, right? So this one is called is polygenic inheritance. Okay, I think you understand. Okay, pleiotropy. We have so far seen the effect of gene on a single phenotype or a trait. There are, however, instances where a single gene can exhibit multiple phenotypic expression. Look, pleiotropic gene means what? When a single gene it express multiple phenotypes, then it is called pleiotropic gene. That is, such a gene is called pleiotropic gene. Which gene is here? A single gene can exhibit multiple phenotypic expression. Such a gene is called pleiotropic gene. The underlying mechanism of pleiotropy in most cases is the effect of a gene on metabolic pathway, and through this metabolic pathway means different phenotype comes, right? Metabolic pathway which contribute toward different phenotypes. An example of this is the disease of phenylketonuria. Which disease is given here? Phenylketonuria, which occurs in humans. This disease is caused by mutation in the gene that code for enzyme phenylalanine hydroxylase. Which enzyme? Phenylalanine hydroxylase. It is a single gene mutation, right? And this manifests itself through phenotypic expressions, which expression characterized by the mental retardedness first, and reduction in hair second, and skin pigmentation third. So 3 phenotypes here we get 3 phenotypes. Which phenotypes? Here skin pigmentation, reduction of hair, and mental retardation. Because 3 different phenotypes are expressed by a single gene mutation, right? And this single gene mutation which codes for enzyme phenylalanine hydroxylase, right? Because a single gene is responsible for 3 different phenotypes, that's why it is called as pleiotropic gene. And this disorder, it is called phenylketonuria. Phenylketonuria. Ketone bodies are expelled out through the urine. Phenylketonuria. Okay. If you have any doubt, you can ask, right? We will solve all the doubts in our app.